Dear students, I am Dr. Akhilesh Kumar, Associate Professor of Botany BCG CW Nagal Chaudhary. Today I am going to speak on the structure and functions of endoplasmic reticulum. Before going to know very detail about this, first we know that who discovered it. There are three eminent scientists who involved in the discovery of endoplasmic reticulum. How it generated, biogenesis of endoplasmic reticulum according to the current concept. Membrane biogenesis is a multi-step process that involves the synthesis of a basic membrane, lipid and intrinsic proteins. Then the addition of the other components such as enzymes, sugars or lipids occur in a semi-sequential manner. The insertion of protein into endoplasmic reticulum is independent of that of the lipids. Some proteins of the endoplasmic reticulum are formed by the ribosomes in the cytosol, which then becomes inserted into the membrane. Just we comes on the structure of the endoplasmic reticulum. Endoplasmic reticulum extends from nuclear membrane at one end and joins plasma membrane at the other end by E or plasma membrane junction. This has been discovered by the OK et al. In 2016. It never opens into cytoplasm. It also continues with other organelles as Golgi apparatus and mitochondria. All these structures, tubules, sacs are held in their place by cytoskeletons. The air remains bound by phospholipids membrane. Inside the membrane, a fluid-filled space exists, which is known as cisternal space or lumen. The lumen is continuous with the perinuclear space. The membrane of endoplasmic reticulum contains many enzymes, triglycerides, phospholipids, and cholesterol. Most important ER enzymes includes esterases, NADH cytochrome, reductase, glucose 6-phosphatase, glycosyl transferases, and magnesium ion activated ATPase. Unit membrane of all cavities of endoplasmic reticulum are interconnected. In electron microscope, the cross section of the endoplasmic reticulum appears as a two parallel membrane separated by narrow light space about 4 nm wide called as cisternae. Now you can see the very clear cut. Thus, in the cell there is a nucleus, and in between nucleus and plasma membrane, these endoplasmic reticulum are present. We can see that clear-cut view of these nucleus and cell membrane, and these are called the endoplasmic reticulum. Component of the endoplasmic reticulum. What are the components by which it is made up of? On the basis of morphology, endoplasmic reticulum is made up of three types of structures. Number 1, cisternae. Number 2, vesicles. Number 3, tubules. All the three components of endoplasmic reticulum are bounded by 50 to 60 angstrom thin membrane. The membrane consists of three layers as plasma membrane: middle, thin, and transparent layer of phospholipids, and outer and inner dense layer of proteins. Fourth one is that perinuclear space is the space present between endoplasmic reticulum and nucleus. There is a close relationship between perinuclear space, cisternae, and cell shape. Just you can see that different components of the endoplasmic reticulum like cisternae, tubules, vesicles, and etc. Just we can how many types? It is divided into two types. Number one is the smooth and two is the rough endoplasmic reticulum. Smooth means there is no ribosomes attached and rough means there is a ribosomes are attached. Next please. Smooth endoplasmic reticulum. It is highly curved and tubular in structure and smooth endoplasmic reticulum occurs mainly in tubular form and does not have ribosomes. The tubules measures actually in size 500 to 1000 angstrom long, forming irregular lattices. Commonly present in cells connected with steroid and lipid biosynthesis and carbohydrate metabolism. Different calls may contain different amount of smooth endoplasmic reticulum depending on nature and function. The smooth endoplasmic reticulum is involved in the synthesis of lipid including cholesterol and phospholipids which are used in the production of new cellular membrane. In certain cell types, smooth ER plays an important role in the synthesis of steroid hormones from cholesterol. In cells of the liver, it contributes to the detoxification of drugs and harmful chemicals. The sarcoplasmic reticulum is a specialized type of smooth endoplasmic reticulum that regulates the calcium ion concentration in the cytoplasm of stated muscles cells and regulate contraction of muscles. Function. There are many functions of the, just you can see that smooth endoplasmic reticulum, transport of proteins, steroid metabolism, already I have seen, release, uptake and storage of calcium ions, and one and last important thing is that lipid metabolism. That carbohydrate metabolism, how it is performed by the smooth endoplasmic reticulum. Just you can see here from the glycogen to glucose and it get phosphorylated and ultimately converted into the glucose and phosphate. Then one and important that function of detoxification of toxic chemicals by the smooth endoplasmic reticulum. These are performed by the various ways. There are involvement of the various enzymes and cytochromes. Just you can see very clearly how this detoxify the toxic chemicals. Now we comes on the rough endoplasmic reticulum. Rough endoplasmic reticulum is characterized by the presence of ribosomes over the surface connected with the production of secretory proteins and is very common in cells involved in growth and differentiation. Rough ER is named for the rough appearance which is due to the ribosomes attached to its outer cytoplasmic surface. It rough endoplasmic reticulum lies immediately adjacent to the cell nucleus and its membrane is continuous with the outer membrane of the nuclear envelope. Rough endoplasmic reticulum is found throughout the cell but density is higher near the nucleus and the Golgi apparatus. Ribosomes are ribonucleoprotein particles present either free in cytoplasm or attached to the endoplasmic reticulum or outer surface of nuclear membrane. Ribosomes are placed for protein synthesis and synthesize structural proteins when present free in the cytoplasm, and secretory proteins when attached to the endoplasmic reticulum. Now we comes on the function of the rough endoplasmic reticulum. Just it is very important because presence of the ribosomes, they synthesize the proteins and help in the transportation of proteins and folding of the proteins. Now we comes on the overall main function of the endoplasmic reticulum. It is concluded that synthesis and translocation of secretory proteins uh, across the rough endoplasmic reticulum membrane. Next point is that integration of proteins into the plasma membrane. Third point is that folding and modification of protein in the rough endoplasmic reticulum lumen. Fourth is that synthesis of phospholipids and steroids on the cystosolic side of the smooth endoplasmic reticulum membrane. Storage of calcium ion in the smooth endoplasmic reticulum lumens, and their regulated release into the cytosol. And last second is that carbohydrate metabolism. Already we have seen the reactions, how these organelles help in the carbohydrate metabolism. And one of the important thing is that detoxification of drugs. Because of the presence of the several enzymes, cytochromes, they helps in the detoxification of the chemicals. Already we have seen the mechanism of the detoxification of the how it helps by the endoplasmic reticulum. Thank you.